So today I'm going to talk to you about a new product. There is actually a new line of products in BioLegend. Now we are trying to introduce new products or new tools that are going to help us to support cell culture. And in this meeting, I'm going to present you this workflow. It's some products that we are developing to support the growing and the culture of T cells. I'm going to have a poster tomorrow. It's a 602 poster where I'm going to present more information. So because of time, I'm just presenting T cells here. But in my poster, I'm going to talk about other products as a cryopreservation product and also results for NK cells. So I'm going to start talking about the cell therapy. So as you know, cell therapy is the use of cells with a therapeutic purpose, and the most common example is the CAR T cells. And also during this therapy, you have to isolate the cells from a donor, and ex vivo, you have to manipulate them. It could be expanding or transforming them, and then you are going to reinfuse them back in the donor. So this process that you do outside is named ex vivo or further bioprocessing. So if you hear that word or that phrase in our products, we are referring to this. All that manipulation of the cells before you reinfuse them. So currently there are some type of cells that are being used for cell therapy. One is the pluripotent cells, some more differentiated adult cells, embryonic cells, but for sure the T cells are the most popular and one of the most promising therapies right now. This is a general picture about the cell therapy for T cells. So everything initiates when you isolate the cells from a donor, but then these cells need to be shipped or transferred into a lab. So for this, you have to freeze your cells in order to keep them alive. And in the lab, you are going to isolate or enrich a specific population that you want. Then you can modify, this could be an lentiviral transduction, and then you have to expand the cells because you are going to need many number of cells in order to be reinfused. Then you have to freeze again the cells to be shipped back to the hospital to be reinfused into the donor. So based on this workflow, we are trying to develop different tools that are going to help to support to keep the cells alive. So in my poster, I'm going to present some data for the cryopreservation solution, and today I'm going to present data about a product that is going to support the expansion of the cells. So currently, people is using AV serum to culture human T cells, and you know the serum is isolated from the blood, and it's been used to support the culture of PBMCs, NK cells, and T cells. And people use it with different type of media. It could be IMDM, RPMA, so it's very flexible. And this is going to provide all the nutrients that your cells need to be alive. These are some other characteristics of the serum. However, using serum, and when you are talking about cell therapy, has many disadvantages. One is that it's containing many undefined components, so you don't really know, you don't have a full characterization what is containing in the serum. And also it could be variations depending of the donor, so depending the source where you isolate the blood. So it could be variations due to the sex, genetics, diet, the health. And also this is gonna have some variations or inconsistencies between lot to lot. So that means that you can have very variable results if you change the brand or if you use another lot of serum. For that reason, people that is using humans AV serum for cell therapy, they have to qualify every lot every time that they use it. Of course, this is going to consume time and also money to be pre-qualifying your raw material. And talking about cell therapy, this is one very important risk that this serum can contain some pathogens. Many of the serums, they have some testing for pathogens, but there are others that are not tested, so you don't know that they are there. And when you are talking about to reinfusing these in a people that is sick, for sure it's going to be a high risk. So that means that if you use undefined material for your cell culture, you are going to have an undefined cell culture quality. And that means that you can have variations and you can have inconsistencies. For that, it's important to develop cell cultures in absence of serum to avoid these variations, and also to have more controlled process and more control and consistency performance. Based on that, we are introducing this new product. It's named Cell-Vive TNK because it's supporting both cell types. Serum-Free because it's a product that is animal-free, and it's a serum substitute. And of course, it's being produced under the GMP regulation. This is some information about our new product. This is a catalog number. You can look for more information in our website. And this is a 20x formulation. That means that you can deliver it at 5% in the media that you prefer. It's very flexible. And it's keeping frozen, well, it's store frozen. Again, it's a GMP manufacturing. And it's for research use only or for further bioprocessing. And as I mentioned before, it's to support T cells and NK cells. Also, we provide some protocols to expand and grow your T cells or your NK cells in the technical data sheet. And part of the quality control and regulation of this product, we are offering the measurement of endotoxin, pH, osmolarity, mycoplasma. We do testing for sterility, and of course we do testing for T cell culture. So now I'm going to show you how our product performs. In order to test this product, we have used PBMCs derived T cells. So we isolate the PBMCs and we culture them in plates coated with anti-CD3 or anti-CD28. And then we culture them in complete media. This complete media is going to be base media. In this case, we use IMDM plus IL-2 to induce the proliferation of the cells. And we are going to have different conditions. One where we use fetal bovine serum, another one where we use the AB serum, the human one, and another one when we are testing our serum substitute. So I'm showing here our results for viability. The first group is for the fetal bovine serum, next group is the human AB serum, and finally we have our Cell-Vive product. And each graph or each color is a different time point. So we measure viability and expansion at day 4, 6, 8, and 11 of culture. And you can see how our product is able to support viability above the 90%, and it's very comparable with the other supplements that you can use to culture your cells. But what about the expansion? When you talk about cell therapy, you need big numbers. So this is the same color code, and you can see how, I didn't mention this, but people is using AB serum because it's better to support the growing for human T cells than the fetal bovine. And you can see this difference here. But when we use our serum substitute, this actually is better and support higher levels of expansions compared with the other supplements that you can use. And finally, we also do a characterization about activation. So here the groups are a little bit different. First, we have the effector cells, then the central memory cells, and then the naive T cells. And each color is one of the different conditions. So the human AB serum and the fetal bovine serum, they are very good to support or to induce effector cells and central memory cells. But our product is actually favoring the development of naive T cells. And why this is important? Well, when it's been demonstrated, there are two advantages. It's been demonstrated that when you use naive T cells for an adoptive transfer, these naive T cells in vivo are going to be more effective in controlled tumors, and they are going to proliferate better than if you use already activated cells. And also, you don't have the risk to be exhausted because they are naive. The other advantage is that you want to use, you can use them ex vivo to differentiate them in the phenotype that you have. So instead of being isolated effector cells or central memory directly from the blood, where you have very low levels, you can actually have your naive T cells expanded outside and differentiate them, so you are going to have a big number of whatever population you want to be used them later. So in conclusion, we have developed this serum substitute that is a non-animal derived product, and is exceptional to perform and to support the viability and expansion of the cells, and is superior than other additives. And also it has the advantage of preserve or favor the development of naive T cells. And yeah, thanks for your attention, and I'll be happy to answer your questions.